Hey guys, and welcome back to SciSci. In today's video, we're going to be taking a look at the light microscope. Now, the light microscope is one of many different types of microscopes, such as electron microscopes. There's two types, transmission and scanning, as well as fluorescence microscopes. But today, we're going to be focusing on the light microscope because it's the most accessible for people who are new to labs. When we do our tour, we should start off with the ocular lens. Now, the ocular lens is what we look through to see our sample. It's usually at the top of the microscope, and it not only allows us to see our final image, but it provides a certain amount of magnification to our image. Paired with the ocular lens is the objective lenses, and these are either above or below our sample. The objective lenses provide the most amount of magnification for our sample, and we move them up and down through fine and coarse adjustment knobs that are found at the base of the microscope. Now, in addition to these parts of the microscope, we have the arm, which either holds the objective or the light source. In this case, it is holding the light source. And this light source either shines down or up to the sample, brightening it so we have a clear view of what we want to look at. The light microscope is commonly used to see organisms such as amoeba. We can see other types of protists, as well as bacteria and plant cells, human cells. Sometimes through a light microscope, we are able to see certain organelles. Specifically in plants, we might be able to see chloroplasts or the main vacuole. And sometimes in animal cells, we may be able to spot the nucleus. Now that we've gone through this quick tour of the light microscope, we can take a look at how it works. So once again, as you can see in this diagram, we have a light source. And this light source shines through the sample. But what was missing in our tour is that we also have a condenser lens, and this condenser lens condenses the light that is shining through the light source and focuses it on the sample so that we have the most amount of light brightening our image. Now, this light passes through the objective lens where it's magnified and passes through a series of mirrors until it reaches the projector and ocular lenses where we receive our final image. Microscopes usually have a series of objective lenses, 3 to 4, and these vary in different magnifications, either from 4 times or 5 times, 10 times, sometimes 20 and 40 times, and as we go to higher magnifications, it may even be 100 times, although that may be more rare. And these different magnifications can be labeled as scanning, low, high, and to better understand how these objectives work, let's take a look at convex and concave lenses. Now, convex lenses have our curves shaped outwards, while concave lenses are curved inwards. And convex lenses are usually what are used in microscopes. So at the top, you can see our little eyeball, and through the eyeball, we have light passing through. The small s is supposed to be our sample. And so when light passes through our convex lens, we can see that the image that is shown is actually the larger S to our eyes because our lens magnifies that image. So typically we use a convex lens in our objectives, and that allows us to magnify the tiny image, either if it's a bacteria or a protist that's underneath the microscope. All right. So now that we've gone through magnification, how light works and how it passes through the microscope, as well as the different parts of the microscope, we've kind of wrapped up our tour of the microscope. If you want to learn more and practice with the microscope, check out SciSci.com, where we have experiments and practice exercises that you can try out on your own with microscopes. Thank you, and I'll see you guys next time.